Chapter 4, Biology of the Cell, Part 2. So now we move on to active transport. Active transport means things moving against the gradient. So this would be, say, things moving from a low concentration area to a high concentration area, or moving against the electrical gradient. So something that's positively charged moving to a positively charged area, or negatively charged moving to a negatively charged area. This will require energy from the cell, and that energy is going to come in the form of ATP. That's right, good old ATP. So one form of active transport is going to involve proteins, membrane proteins. So for instance, we have the ion pumps. Ion pumps will move ions, move them against the concentration gradient. So for instance, erythrocytes, red blood cells, need to send calcium ions out of the cell. And it takes energy, the use of ATP, to move charged calcium ions out of the cell to outside of the cell where it's at a higher concentration. An example also is the sodium-potassium pump. This is sort of an ion pump, except it's an exchange pump because it's moving materials in two directions. So, for instance, we have sodium-potassium pump has ATP bind to it, and then that ATP will dephosphorylate, becoming ADP, and this will allow sodium ions to exit the cell to go to the fluids outside of the cell where there's already lots of sodium ions. And then potassium will be able to bind to the sodium-potassium pump, and then as the phosphate group is released, it will allow the potassium ions to enter the cell where there is lots and lots of potassium ions, and then the sodium-potassium pump will be ready to go through this process again and again and again. So 3 sodiums leave the cell, and then 2 potassiums enter the cell, and this requires ATP to work. This is extremely important in our bodies because it is critical for the function of neurons. Then there's secondary active transport. This is where the protein is using the energy caused by something moving down its gradient to allow something else to move against its gradient. So for instance, we know there's lots of sodium outside of cells, less sodium inside. So this secondary active transport allows sodium to go from high concentration to low. It can also use that energy to allow glucose to move into the cell, going from low concentration to high because we want to hoard the valuable nutrient glucose. Since both things are moving in the same direction, is called a symporter because both things are going in the same direction. Surprise, surprise, there's also antiporters where something like sodium ions might be moving in via the concentration gradient, and then something else, say hydrogen ions, is moving out, going against the gradient. And that would be antiporter. And again, this is using kinetic energy to drive the movement of something else. So technically, ATP isn't involved. We also have vascular transport. This is using vesicles to move materials. A vesicle is a membrane-bound structure. It's like a sack or a bag filled with something. And the production and movement of the vesicles requires a lot of energy. So you won't see ATP noted anywhere, but a lot of ATP is being used. So exocytosis. Exocytosis is where things are produced inside the cell in a secretory vesicle. Then that vesicle will move to the plasma membrane, fuse with the plasma membrane, and then expel its contents outside of the cell. So exocytosis, things are exiting the cell. They were formed and packaged in a vesicle and then sent to the plasma membrane to be released. So for instance, neurotransmitters released by nerve cells. There's other things engaging in exocytosis. Then there's endocytosis, bringing things into the cell. This is when the cell wants to bring something big or a lot of something into the cell. So the plasma membrane will actually form an invagination and eventually pinch off, forming a vesicle inside of the cell filled with whatever it is it wanted to bring into the cell. There are 3 main forms of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So phagocytosis, these cells' plasma membrane forms pseudopodia, which surround a food-like, solid-like particle that the cell then will bring in to the cell in this lovely newly formed vesicle. So this is phagocytosis, sort of like cell eating, and then a lysosome will fuse with this vesicle and release digestive enzymes to break down that large solid material. So for instance, our white blood cells do this when they surround and bring in bacteria to destroy. Then there's pinocytosis. Pinocytosis is sort of cell drinking. So the cell will pinch off and form a vesicle filled with fluid that is surrounding the cell. So it's taking a little sip, bringing in the fluid with the solutes dissolved in it into the cell. And then finally, receptor-mediated endocytosis. So this will include special receptor proteins that are part of the plasma membrane. The receptors will bind to a specific ligand, aka a specific molecule of some kind. And when enough of the receptors have bind to their ligands, this will trigger the formation of the vesicle, bringing that important material into the cell. So when, for instance, cholesterol is often brought into cells through this process, receptor-mediated endocytosis. Now it turns out viruses can take advantage of this, and a virus can have the spikes on the surface of the virus look a lot like the ligand that the receptor wants to bind. So the virus will bind to those receptors and trick the cell into bringing the virus into the cell, then allowing the virus to take over and turn it into a virus-producing factory. So for instance, influenza, the flu virus, uses this approach to get into the cells lining our respiratory tract. Viruses on the surface, virus being brought in, virus all happy in its little vesicle. Here is some summaries from the textbook. They're pretty awesome, covering the basics of these various forms of transportation, either in picture form or in lovely table form. So definitely take a look at those in the textbook. All right, cell communication. Cells need to communicate with each other. Some of this communication is directly by touch, cells touching other cells. So, for instance, our immune response cells come up and touch different cells looking for the invading cells, the pathogenic cells. So anything unhealthy or not self, the receptors on the non-immune response cell will bind to the unexpected surface molecules of that cell and destroy it. But if the immune response cell finds the expected surface structures, then it won't destroy it. It'll say, oh yeah, you belong here. You're supposed to be in the body. And basically, it is those carbohydrates forming what's called a glycocalyx that the immune response cells are checking by touch to determine if a cell is supposed to be in the body or not. Sperm and eggs have the sperm has proteins that will bind to the unique carbohydrates on the surface of the egg cell that will allow for fertilization to occur. And then cellular regrowth following an injury, cells will make contact with each other and try to fill in the spaces where there aren't cells. They go, wait a minute, Bob's not here anymore. Bob's supposed to be there. I know, I'll divide and produce a new Bob. So cellular regrowth. And then eventually they'll fill in the spaces, make contact with each other, and say, good, we are done. This space has been repaired. And then, of course, communication can be from the distance. Think of it sort of like as mail coming in. So ligand receptor signaling, very common. One cell will produce ligands like hormones, and it'll travel through the bloodstream and reach other cells, and those other cells will have receptors that bind to the ligand. So one cell produces the molecules, the other cell has receptors that will bind to it. Three main kinds: channel-linked receptors, enzymatic receptors, and G protein-coupled receptors. Channel-linked receptors. These are proteins that are transmembrane proteins embedded in the plasma membrane. They have a binding site for the ligand, and when it binds, it causes a channel to open, allowing something to pass through, often ions, then flooding into the cell from high concentration to low concentration. Again, very important for neurotransmitters, for moving that electrical charge along muscle and nerve cells. Enzymatic receptors. In this case, the ligand will bind to the receptor, and what will happen is at the other end of the receptor, inside the cell, facing the cytoplasm, an enzyme will activate, causing enzymatic activity. So the receptor is also an enzyme that's inactive until the ligand binds. And then G-coupled protein receptors are a slightly more complicated version of the enzymatic receptor. Here the ligand binds to the receptor. That protein activates and releases a G protein that goes out and causes something to happen. It may cause channels to open. It may cause enzymes to become activated. So the G protein is going to be a messenger inside the cell. So it's released when the ligand binds and goes out and does something specific that's necessary for the cell. All right, we also have structures of the plasma membrane. These are extensions of the plasma membrane. They include cilia and flagella. Cilia and flagella, both projections from the cell. They are both made up of the plasma membrane as well as microtubules. So microtubules form the core of the flagellum, form the core of the cilia. Microtubules, long protein filaments, part of the cytoskeleton. And the cilia, when it's found on a cell, you find lots and lots of cilia on the surface of the cell that is exposed to space. So the exposed surface of a cell may have lots and lots of cilia on that surface. And the job of the cilia is to beat, is to move, is to generate a current, to move things outside of the cell. So cilia move things outside of the cell. Here you can see a cross-section of a cilia with all those microtubules inside of the cilia. And the cilia is found in cells, for instance, lining the respiratory tract. In that case, it's moving mucus. Flagellum. Flagella is much longer than cilia. And if a cell has one, it usually only has, if a cell has flagella, it usually only has one. So really we should say flagellum. And the job of the flagellum is to propel, to move the entire cell. There is only one human cell that possesses a flagellum, and that is sperm. Microvilli are another example of extensions of the plasma membrane. In this case, these are somewhat smaller extensions than we saw with cilia. Again, exposed to a surface, a space. The microvilli are densely packed, so there's tons and tons of them on the surface. And they are formed by microfilaments. Microfilaments are also long protein filaments that are part of the cytoskeleton. They're a bit smaller than microtubules. And the point of the microvilli is to increase the surface area of the plasma membrane. So now there's lots and lots of more surface area of the plasma membrane on this side. And that is ideal to increase absorption, to increase the movement of materials from outside the cell through the plasma membrane into the cell. So for instance, small intestine, they have lots and lots of microvilli to increase absorption of nutrients into the body, into the bloodstream. Here's just some electron microscopic views. So here's just the plasma membrane, the microvilli, and the glycocalyx. Plasma membrane, here is the microvilli shown glowing. Here's the glycocalyx, again, the carbohydrates on the surface that let our immune response cells know that they belong here. And then here's just the microfilaments filling up the microvilli and then going out to be part of the cytoskeleton. Lots and lots and lots of little microfilaments. Pretty cool. All right, membrane junctions. They hold cells together. We have tight junctions. Tight junctions encircle, go completely around the circumference of the cell, and they hold those cells together. Tight junctions act sort of like superglue, fusing the plasma membranes of neighboring cells together, preventing things from passing between the cells, such as digestive enzymes. Desmosomes. Desmosomes are isolated proteins in packets that help to connect cells and keep them held tightly together. They're anchors. They're like rivets. They resist stresses like found in the external layer of the skin. Hemidesmosomes are not quite desmosomes. They're half a desmosome, and they connect the cell to the basement membrane. Basement membrane found in epithelial cells only. So desmosomes between neighboring cells. Hemidesmosomes with the basement membrane. We have gap junctions. Gap junctions are where proteins form a channel between the cells, allowing fluids to pass between the cells. And that is it for this lecture.